Hello, this is David Schindel. I'm Executive Secretary of the Consortium for the Barcode of Life, or CBOL. This is the first presentation in Phase 2 of the project's training activities. We will be scheduling a consultation with members of two working groups, the informatics leads and the specimen standard operating procedure working groups. The information that goes along with this project is all stored on Google Drive. If you don't have access to Google Drive, please set up a Google account. You don't have to have a Gmail account, but you do have to have a Google account. You can use any email address for it. And then we'll give you access to this drive and all the folders in it. I want to point out in particular Phase 2 training folder highlighted here. It has all of the materials you'll be seeing. The overall framework, the overall outline for training you'll be receiving is in the highlighted document, BWP Training Framework. So please take a look at that. The goals today are to begin the process of learning about the specimen and data workflows that we'll be using in the project. Now, many of you are experienced barcoders, and we don't expect you to start from scratch. But the project has very high quality requirements. Data that are going to be presented in court have to have much greater reliability and traceability than data that we've used for academic barcoding projects. So even if you've had a lot of experience, we hope that you will take to heart the need to improve your skills, and that's what this training is for. So some of you are beginning at the very start, and this will be an education. Others that have more experience, we're just really going to be looking over your shoulder, pointing out some improvements, so we hope that you'll go along with that. We've asked you to share a lot of information about how you're already doing barcoding, and we thank you for providing us with that information. It's helped us plan this training, assess where you are in the learning process, and to tailor the training for each participating lab. We'll be introducing hardware, software, and informatics processes. We also, in this presentation, I'll begin the introduction of the barcode data standard as it's going to be used for BWP. Now, most of you, maybe all of you, are familiar with the barcode data standard, but the project is going to be adding to the standard, so the data that we're supporting production of is going to meet a higher standard than you're used to in the past. Step 2 in the training sequence is going to go into much more greater detail about specimen informatics, and this presentation will set the stage for those. Let's take a very high-level view at what we're doing. The BWP process begins with identifiable organisms and crime scene exhibits that can't be identified, things like bushmeat or powders. So the identifiable organisms could be specimens in nature living in the wild. They might be living in captivity like zoos or botanical gardens, or they might already be dead and preserved in biological repositories. So that's the input. That's where we begin, the material we begin with. Then we apply standard operating procedures for collecting, curating, and processing these specimens in the lab. And what we wind up with, the outputs, are curated voucher specimens in museums and herbaria, frozen tissue specimens and DNA samples in freezers, in frozen collections in repositories. We'll have databases of specimens and samples. Then we'll have the barcode records in GenBank, all of which together can be used as evidence in court and for expert testimony in court. So those are the very fundamental inputs and outputs of the project. Well, how do we go about this? For collecting organisms, the partner countries are going to be providing the collecting permits and the permissions to take samples, and they have to have standard operating procedures for humane treatment and for the proper preservation of the samples. CBOL will provide you with collecting equipment, funding for bags, tags, cameras with GPS data capture. We're going to be providing you with template spreadsheets with the core data fields that are required for barcode status in GenBank and for the barcode data that we need for this project. That's a minimum set of data fields. You'll probably have other data that you want to collect for your own research and as standard data elements that your collections require. We'll be providing you with supplies for capturing the data, capturing the tissue or fluids, vials, preservatives, labels, all that material will be provided to you. Once the specimens become collections, we're counting on the partner countries to provide the facilities, the cabinets, the buildings, the environmental control, and also management policies for preserving the collections, the security, the environmental control, access, who is allowed to borrow the specimens, who's allowed to access them, in the form of standard operating procedures for curation, for preparation and curation of collections. CBOL will be providing you with funds for storage equipments, could be cabinets or freezers. We'll be providing you with funds for the supplies you need to preserve and curate specimens and tissue. For the tissue that goes into freezers or holds tissue while they're being processed, you'll be provided with labeled vials and barcode readers for the vials for data management, and also the sample management software that's used to read the barcodes on the vials. So for many of you, these will be new systems. We hope you like the ones that we've selected. They are manufactured by a company called FluidX. These are examples of the vials we'll be using. They fit in racks which have barcodes. This is a single barcode reader for a single vial. You'll notice that there are barcodes on the sides of the vials and 2D barcodes on the bottom. And you'll be learning more about this during the subsequent training. Now, when we finally get to the information stage, we've turned specimens into collections. Now we're turning collections into information. We're going to count on the partner countries to provide management policies for your collection data. That is how you keep track of your collections. And we hope that you'll develop draft SOPs for managing the barcode data in your own laboratories. We're also counting on you to have collection management database software. That is, your museums and herbaria should have digitized records of the specimens in the project that would become part of your collection management system as a whole. CBOL will be providing guidelines for meeting the barcode data standard and also a software validation system so that you can take your specimen data, submit it as batches, and have our data quality validation system check that all the required data are there and seem to meet the quality control tests. We'll be providing you with field information management systems. This is what you use to preserve, to store and manage the data that you bring back from the field in spreadsheets. The field information management system can then push the data out to your museum catalog system and also into the Genius LIMS, the laboratory information management system that we'll be providing you with. Some of your institutions may not have collection management software systems. If you are interested, we'll be happy to recommend some and help you start implementing and installing a collection-wide data management system. We're also going to be providing you with what we call the BWP bookshelf. It's another folder on Google Drive inside the Phase 2 training folder. There are 2 folders in there, and they provide you with resources, published resources on protocols. So the first one is the open access resource bookshelf, and these are published and unpublished guides to protocols, workflows from museums, other laboratories, published procedures for barcoding, whole book chapter on preserving specimens in the field for subsequent molecular analysis. Lots of published material and unpublished material that you'll find useful. That's in the open access bookshelf, and all of you will be given access to that. There is one folder with more restricted access, and that's because it has a copyrighted book in it. We have purchased for each partner country one legal copy of this, and we're posting it on the bookshelf, and we are asking that, and we will provide everyone receiving training with access to this. We ask that you do not download or forward copies of this or post this PDF on the bookshelf, on a website somewhere. So each project manager, each partner country has a project manager. We will rely on the project manager to send us the email addresses of all the trainees who should be given access to this folder. Those addresses should be sent to Stephanie Wezowitz. She's the administrator for CBOL, and she'll set those people up with access to the big barcoding book that was edited by Kress and Erickson. Now let me shift gears. I've talked about the extension of the barcode data standard. This is a picture of Peter Christie. He's a vice president at Life Technologies in San Francisco. He's in fact in charge of their capillary electrophoresis departments. And at the Adelaide, Australia Barcode Conference in 2011, Peter organized a session with government agency representatives and private companies for a discussion about why aren't regulatory agencies and companies adopting DNA barcoding? Why is most of the barcoding being done by academic taxonomists, and why weren't other organizations adopting it? And the answer came back very clearly that people in regulatory agencies and companies didn't yet trust barcode data in GenBank. Now, this is the barcode data standard that was created by the database working group in CBOL in 2005. Bob Hanner at the University of Guelph was leading the working group then. And it's meant to be a system for requirements, data requirements in GenBank that would ensure reliability. And the people at Peter Christie's session said, yes, they have a lot of faith in the sequence data. It was the taxonomic identifications that they didn't know if they could trust. So let me review what's in the barcode data standard and how we're going to address this shortcoming. So the barcode data standard sets out a set of required elements for data records that are submitted to GenBank and will be given the reserved keyword barcode in uppercase letters. Now, we chose GenBank to ensure that the data would remain available for a long time, and having one large database made it possible to compare data records from a single approved barcode regions. It would guarantee that the records with the barcode keyword had some minimum quality standard for the sequences. Since georeference data are required, it would enable the data to be mapped, provide traceability back to the voucher specimens, and provide users with direct access to the raw data from the sequencer in the form of the chromats. And we thought that this would pave the way for regulatory and forensic use in court and also for commercial use. But as Peter felt, regulatory agencies and companies weren't adopting barcoding, and we found out why when we asked them. So here are the required elements. There has to be a taxonomic identification down to the species level. There has to be a voucher specimen ID in a standard format. The record in GenBank has to include the name of the barcode region. There are certain requirements for the length of the sequence and the quality of the bases, and the forward and reverse trace files, as well as primer sequences and geographic origin down to country, ocean, sea of origin. Now, note that we didn't require latitude, longitude. I'll talk about that in a minute. The voucher ID in GenBank for barcode keyword status had to be in a formal format. It's a three-part format: the institution code, the collection code, and the catalog number. So NHM UK is the Natural History Museum in London. ENT is their entomology collection. 1, 2, 3, 4, 5, 6 would be a catalog number. For specimens that haven't been accessioned into a collection yet, into a formal institutional collection, you could have field numbers. So we call the institution in those cases personal. That's the generic name for personal collections. Some unique identifier for the person who made the collection, in this case Dan Jansen, and then their field number. Either of those types, either an institutional collection record or a personal collection record, are acceptable. We also had several highly recommended elements. Latitude and longitude were recommended but not required, and that was for two reasons. First, many of the specimens in legacy collections, pre-existing collections, predated GPS, so we don't have latitude, longitude for them, but we wanted to be able to include their barcodes. For protected localities of endangered species, you could publish the latitude and longitude down to minutes and leave seconds out, for example, or degrade the precision of the latitude longitude to protect the locality for endangered species. We recommended the name of the collector, the date of the collection, and the name of the identifier, but we didn't require these in the regular barcode data standard. For the taxonomic identification, we required a genus and species in a combination that could be found in a published source like the Catalog of Life or the Zoological Record or the International Plant Names Index, or in a taxonomic treatment such as a taxonomic revision, or a published description of a new species, or even a provisional label for a potential new species. All of those would be acceptable as taxonomic identifications. So here's what a barcode record looks like in GenBank. In blue you see the keyword barcode. Down here is the voucher specimen, and it's in the form of USNM: birds: 607559. Now in this case, that specimen voucher is hot linked directly through that triplet, the Darwin core triplet, to the specimen catalog at the U.S. National Museum. So you can click on it and jump directly to it, to the specimen record, thanks to what we call a specimen resolver web service. And only a few organizations have these, but more are coming online. Now, each of you will be asked to submit voucher specimens data in this form, which means you have to know the collection code, or the institution code and the collection code for the repository where you're keeping your specimens. Up until a few years ago, there was no standard source for institution codes and collection codes, so CBOL created biorepositories.org as an online registry of institutions and collections. But we found that there were a lot of ambiguous ones. So for example, AMNH, I think of generally as the American Museum of Natural History, but it's also the AMNH institution code is used by the National Herbarium in Iceland. And we found that there were lots and lots of these institution codes that were being used by more than one institution. So We tried to improve the system by merging data with Index Herbariorum and Biodiversity Collections Index, another registry, into one. And now there's a single source of information, the Global Registry of Biorepositories, or GRBio. It's at grbio.org. CBOL has developed and operates this. It has 7,000 biorepository records, records of collections within them, personal collections, and we've resolved this problem of multiple use of the same institution code with a simple device. Most of the double uses were, as I mentioned, between herbaria in Index Herbariorum and zoological collections or natural history collections. So what we've done is taken the institution codes like AM and H, and we've added bracket IH bracket to show that this AM and H is a herbarium in Index Herbariorum, and this AM and H is something else. So we were able to take the number of ambiguous IDs down from 299 down to 128. So now there are less than 4% of the institution codes that are ambiguous. So let's go back to the barcode record in GenBank. Here we have the DBX ref to a taxon that's linked to this species name. That takes you to the taxonomy browser of NCBI and a whole group of resources about taxonomic information of that name. We also find that there are a total of 27 other nucleotide records for this species, other gene sequences, gene regions, other than the barcode region. So here they are. Some of them are barcode records, some of them are other gene regions. On the barcode record in GenBank, you'll also find a link to the trace archive. If you click on that, it takes you to the electropherogram, the trace files for these, and you can go back and check the base call on any particular position in the barcode region. Now, barcoding began with the proposal in 2003, but the barcode records didn't really start to accumulate in GenBank until 2010. So the barcode standard was started in 2005, but the real increase started in 2010 with IBOL, the International Barcode of Life Project at Guelph. And there was a very rapid increase in the number of records that were given the barcode flag. It's worth pointing out, many people think that CO1 as the barcode region for animals is not as useful as cytochrome B, Site B, that was used previously. This is the historical trace of cytochrome B, which is the number of records shown in red, and CO1 shown in blue. The dark colors are records in GenBank with a voucher specimen, with some kind of information about a voucher specimen. The lighter ones are without vouchers. So up until 2006, there were about the same number of vouchered Site B and CO1 records in GenBank, but after that, look what happens to the number of specimen records, voucher specimen records for CO1 versus cytochrome B. So now there are 5 to 10 times as many barcode records with voucher IDs as cytochrome B. So we can see that barcoding has really generated a lot of data. But how well are we complying with the barcode standards? So what we have here are the barcode records in GenBank, the number of records with something in the voucher ID, some entry under voucher specimen ID, and the number of records with latitude and longitude. So for barcoding, there's about 350,000 barcode records in GenBank, of which almost all of them have something in the voucher specimen field, and 83% have latitude longitude. Now, if you compare that with Co-1, all the Co-1 records, barcode and non-barcode, you'll see that there's over 400,000 of those, but the percent with, that is Co-1 that are not barcode records, there are 400,000 of those, slightly more than the barcode region, but of those, less than half have any information on the voucher specimen, and 20%, only 20% have latitude and longitude. If you look at 16S, there's a huge number of 16S records, but only 3% of those have any information about the voucher specimen, and only 9% have latitude longitude. Cytochrome B, as we showed in the last slide, 240,000 records, but 35% have any voucher specimen information, and only 3% have latitude and longitude. So it seems like we're doing a good job of complying with the barcode data standard, but let's take a closer look. There are about 2.6 million records in bold, but only about half of those have been made public. Of those 50%, of those 1.3, only 350,000 have been submitted to GenBank and have gotten barcode status. And as I said in the last slide, about 100% of those have something entered in the voucher ID field, but only about half of them have a formatted voucher ID. So a lot of those records have voucher IDs that really can't be interpreted. They don't lead you to a collection or an institution. And less than half, or I'm sorry, about half have SP in the taxonomic identification field. That is, they don't really meet the standard for an unambiguous species name. So, how effective have we been in meeting the expectations of regulatory agencies and companies? Well, not very, and that's because of this weakness in the voucher IDs and in the taxonomic IDs. Several years ago, Rod Page, a leading biodiversity informatics specialist at the University of Glasgow, noted the rapid increase of what he called dark taxa, that there was an explosion of data records in GenBank that didn't have names. And these were eyeball records that only had the name of the order and bin number, and also all the things that had SP in them. So at that point, in 2010, Rod found that there were not that many barcode records that had formal taxonomic names that appeared reliable. The U.S. Food and Drug Administration had a similar reaction. They adopted barcoding several years ago as a tool for identifying seafood, but they wouldn't use barcode data in GenBank. And it was because they wanted their data to be, their voucher specimens to be authenticated. That is, they wanted for each of their voucher specimens, they wanted a taxonomist to say, I've looked at this and I've identified it. So their reference fish encyclopedia, which is online and separate from GenBank, only has 427 records and 389 vouchers, but each of those is authenticated. That is, they have a piece of paper from a taxonomist that said, I identified this. These are not barcode records in GenBank, and they're not in bold. So there's a separate database that FDA has developed because they didn't find the voucher specimen ID or the taxonomic IDs of GenBank barcode records reliable. So, what have we learned? That a significant number of the barcode records in GenBank don't comply with the barcode standard. Now remember, many of those were put into GenBank early in barcoding before we really started to enforce all of the requirements. So we're now going back and asking people to fix their voucher IDs. Even in the case where the voucher ID is properly identified, there may not be enough reliability to the taxonomic identification for regulatory and law enforcement agencies to use them. So this indicates that we need additional data and metadata that make the taxonomic identifications more reliable and more transparent. So that's what we're doing in BWP. We're going to require all the usual elements of the barcode data standard, plus the recommended elements that I mentioned before, and we're creating 5 new metadata fields related to the reliability of the species identification, and we're going to add 1 demarcation to set our BWP records aside from barcode records, and these will be barcode platinum, barcode gold, and barcode silver. So, in addition to all the usual requirements for the barcode data flag in GenBank, we're going to ask you all to record the name of the identifier, the person who made the species identification, the date of the identification, the type status of the voucher specimen, that is, if it's a holotype, if it's a paratype. Most of them won't have any type status. And 2 very new fields, basis of identification and the confidence level of the identification. What do we mean by basis of identification? Now, these are data records, metadata fields that we're going to be developing together. These are CBOL's initial recommendations, but these are going to be open to change and input with your help. So, under basis of identification, you can enter several different values. One will be, what is the material you're looking at? Did you make the identification based on a complete adult, or was it a partial adult? Was it a juvenile? Did you make the identification based on a digital image, not the specimen itself? Or did you just use a tissue sample? Second, what did you compare it to? Did you look at reference specimens in a collection, or were you looking at descriptions and images in a taxonomic work in the literature, or were you looking at a field guide? So these are the kinds of information, qualitative information, that the user can look at and say, okay, well, now I understand how the identifier identified this as a member of that species. What about the levels of confidence? This is a difficult and subjective appraisal we're going to ask people to make. The highest level of confidence would require that the taxonomist who made the identification, and remember their name is going to be on the record, that the person who made the identification is a professional taxonomist with some experience in this taxonomic group, that the identification was made on a complete specimen or a digital image of the specimen, and that the level of confidence in the species identification is high. That is, the person asserts there's a high probability that this species identification is correct. Then we are going to have medium level of confidence, that it is an experienced amateur or a professional who is not a specialist on this group, and for whatever reasons there's a high probability that the genus identification is correct, but not so high that the species is correct. And then we'll have a third level of confidence, which is just marked preliminary identification, when there's not a high probability that even the genus identification is correct. Barcode platinum records are going to be those that are based on voucher specimens that were collected and analyzed under chain of custody. That is, they were placed in tamper-indicative bags with unique identifiers, that there's an audit trail showing change of responsibility of who had control, physical control over the specimen, and that it was placed in secure storage with controlled access, that it was processed in a police lab, and that the specimen identification has the highest level of confidence, as well as all the required elements in the barcode data standard. Barcode gold records are those that are based on tissue samples that were managed under chain of custody, so that the DNA are going to be identical to a platinum record, but they were processed in an academic lab. And here again, we're going to require a highest level of confidence for a barcode gold record status. Finally, barcode silver are the great majority of specimens. Those are based on scientific samples not collected under chain of custody, that they were barcoded in an academic lab, but that the taxonomic identification has the highest level of confidence. This is the Barcode of Wildlife Project's website. It's barcodeofwildlife.org. And if you go to that page, you'll see on the navigation bar priority species. Siebold has compiled your lists of priority species, and there are now 774 endangered species distributed among the following groups. These are for the 4 countries in different colors. The blue bars in the background are all 4 countries taken together. And you'll see that plants over here, plants are almost 400. So roughly half of them are plants, and the others are distributed invertebrates, fish, reptiles and amphibians, birds and mammals. There are some noteworthy high points. So for example, there are a lot of reptiles and amphibians on the priority list for Mexico. Nigeria had a particularly high number of mammals on their list. Most of them, this is the distribution according to the protection status: CITES Appendix 1, Appendix 2, Appendix 3, and not in CITES. And you'll see here again countries going from front to back, totals in the back, roughly equal number covered by Appendix 2 in CITES, and those not in CITES but protected by national law. So that's very interesting how international trade regulated by Appendix 2 and domestic endangerment by domestic trade have about equal representation. Number of barcodes already in GenBank, the vast majority of them have none. So there are some, a few in different countries that might have 1 to 4. Very few have 5 to 9. Almost none have more than 10 barcode records already in GenBank. So we have a lot of work to do. We've also created a species viewer. If you go to Barcode of Wildlife and under priority species, you'll see this viewer. It's a very handy way of browsing through. Groups, taxonomic groups are in colors, so plants are dark green, invertebrates I think are red. Also the countries that have listed them, their flags show here. And in Roman here you see the appendix, the CITES appendix in which they're listed. You can sort them in different ways. You can filter things out to drill down to the group or country of interest. And when you click on an individual tile, you'll get access to the Encyclopedia of Life, an image from different sources, link to the NCBI taxonomy browser, barcode records in GenBank, GBIF records for specimens, the IUCN page for that species. And if you have other sources of information you'd like integrated into the species browser, we'd be happy to talk to you about that. So that brings me to the end of this introduction. We will now be scheduling webinars or go-to-meetings with you all. We'll be contacting your program managers to help coordinate those dates and times. You're free to do them as an entire group, that is everybody in the partner country together, or it might be that you want all the informatics leads to do it together, or all the specimen SOP people, or we could do it one lab at a time. You should discuss that to see how you want to proceed with those consultations. Thanks very much for listening, and I look forward to taking these first steps in with consultations with you.